Hello, I'm Professor Matthew Schmidt, and this is Genetics. In this session, we're going to discuss, in our general exploration of DNA structure and replication, the structure of DNA. If you're following along in order, last time we spent a bit of effort going through the experiments and the rationale of determining, in fact, that DNA was the genetic material. This, we had said, was pretty much confirmed by about 1952. And then people got very interested in DNA. And you probably know that this ultimately culminated in Watson and Crick's Nobel Prize winning discovery of the exact structure of DNA. It's very important to remember in all of biology and even in other things that structure and function are so intimately related. So in this case, we understood the function of the gene of DNA much more than we understood the structure to begin with. But the structure, in other words, they're not separable. We mentioned certain characteristics that a molecule would have to have in order to be the genetic material, and the structure of DNA that Watson and Crick ultimately elucidated explains how this wonderful molecule can do that job. So before we get ahead of ourselves, the last question that we asked when we were talking about it last time was, what was biochemically known about DNA? Meaning, at the time in the early 1950s, right before Watson and Crick, I had mentioned to you that biochemists were aware of the existence of DNA and knew certain things about its general structure. It was known to be present in chromosomes, but it was thought to be a relatively unimportant just a molecule that was part of the fabric of the chromosomes, not having to do with information. So what was in fact known at that time was that DNA, like so many biological molecules, was a polymer, which means that there are subunits that are strung together into, in this case, a very long line. The subunits are known as nucleotides in this case, and there are 4 types of nucleotides, which we're about to get into. Just remember, originally it was thought that protein was the genetic material. This is not the only reason, but it was in large part because there are 20 different amino acids from which to create variation. We only have 4 here, but it turns out that you really don't need a lot of different different types of subunits to encode a ridiculously large amount of information. So there are 4 types, and nucleotides, as monomers or subunits go, are relatively complex. So each nucleotide has a sugar, and in the case of DNA, that sugar is called deoxyribose. It's a 5-carbon sugar. There's a phosphate group, and there's also a nitrogen-containing base. So let's look in a little bit more detail at this. So we did say that there are 4 different bases, but my point here is that it is the base and the base alone that causes one nucleotide to differ from another. In other words, all 4 of the different nucleotides are the same in the sugar and the phosphate group part. They differ only in the base part. So 4 different bases allow us to have 4 different types of nucleotides. Those 4 are divided into 2 categories based on the size of the base. So purines, so-called, purine bases contain 2 rings. I'll show you a picture in a moment. And those bases are adenine and guanine, often abbreviated as A and G. The pyrimidine bases only have 1 ring. They're literally shorter in length, which is important in the overall structure. Pyrimidines have 1 ring, and those are cytosine and thymine. And as you may know, those are often abbreviated as C and T. A lot of times when you're dealing with DNA sequences, we want to just write it as C-A-T-T. We have to know what we're talking about, but a lot of times the abbreviations are used. However, for right now, we want to go into a little bit more detail because the base itself is named either adenine or guanine, cytosine, and thymine. But when the base gets attached to the sugar, first of all, this is how they're built, is that's called a nucleoside. And then when the phosphate group is added on, that's when they become a nucleotide. Many of you may know this, but I know I didn't know it for a long time when I was a student. The names change when the base is incorporated into a nucleoside or a nucleotide, and the names change in the following way. So a nucleotide containing the base adenine is now referred to as adenosine something, which we'll get to in a minute. And it's a good idea to, I was going to say memorize, I'm not positive if that's the word, but it's certainly good to recognize if you see adenosine, you know that they're talking not just about the base. The base is always adenine. Well, in that case, if the base is guanine, the nucleotide built, or I should say that contains that base, is going to be guanosine. So those two, adenine and guanine, they change in a similar way to, instead of nine, they change to ozine. The cytosine and thymine change in a little bit of a different way. Cytosine becomes cytidine if it's in a nucleotide, and thymine becomes thymidine. Now, I want to show you pictures of these and talk a little bit more about the naming conventions. But before we leave this slide, I think it's very important to, a lot of people ask, how can I easily remember which are the purine bases and which are the pyrimidine bases? And it turns out there are a couple of good ways to remember. If you think of one on your own that works, that's great, but I have two for you. One of them is that the word pyrimidine has a Y in it, right? And both the bases cytosine and thymine also have Y's in them. You'll note that none of either the word purine or the purine bases have a Y. So some people like to think if I see Y, that means the Y's go together in a sense. Cytosine, thymine, pyrimidine. Another way that focuses more on the purines is to remember this saying. It's a little silly, but a lot of these sayings are. Pure as gold. If you think of gold being a pure metal, pure as gold refers to not only the P in the purine, but the word pure is in purine, as for adenine, and gold for guanine. So both of these are going around in my head. You can use both of them. You can pick one, or as I said, you can make one up yourself, but you're not going to get better than this. I think you are expected to know which are the purines and which are the pyrimidines. So figure out, use one of these ways or both. Now, let's look at some excellent pictures of the full nucleotides, talk a little bit about it. Now, first of all, so often the nucleotide is depicted this way, but you'll notice that there's one phosphate group here attached. So these are monophosphate versions of the nucleotides. It turns out that there can be, I'm not going to draw out the whole phosphate group here, but I'm just going to write PO4 to mean a phosphate group. There can be two of them stuck on, in which case this is a diphosphate version, and there can be even three of them stuck on. The chemistry isn't exactly correct, but then you would have, with three, right, a triphosphate situation. So nucleotides may be monophosphate, diphosphate, or triphosphate. When they're in the pool of nucleotides in the cell, they tend to exist as the triphosphate version, and when they get incorporated into the chain, two of the phosphates are used in that incorporation. They're used up. So in the chain, they exist as we've drawn it here as a monophosphate nucleotide. So let's look at the naming conventions here. So the base adenine is right here. You can see that there are two rings there. It's a purine, right? Also, this is it's biochemistry, but it's not a bad idea to remember that the base is always attached to the 1' carbon of the sugar, whereas the phosphate group is always attached to the 5' carbon of the sugar. We often hear about the 5' and the 3' ends of DNA, and that's because when the bonds are made, this is not what this picture is really meant to show, But if these nucleotides were going to be put together into a polymeric chain, again, the chemistry isn't exactly right, but a bond would form, a phosphodiester bond, between the phosphate group of the incoming nucleotide and the OH group that's on the 3' carbon of the sugar. So what that means is, think of this as just a little tiny chain. There's always a free OH at the one end, which is the 3' end, and there's always a free phosphate group at the other end, which is known as the 5' end. I don't want to go too far into that. It gets into biochemistry, but I think it's a reasonable amount of detail to know. Now, back to the naming convention. This nucleotide here that contains adenine, because there's one phosphate group on it, would be referred to as adenosine monophosphate, or AMP. And you may have heard of the molecule cyclic AMP, or some people call it CAMP. It's a very famous nucleotide-based signaling molecule that's present and does a lot of functions in cells. So my point in mentioning that is that sometimes nucleotides have other functions besides genetic ones, which implies a few things, but nucleotides have been around for a long time, let's just say that. Down here we have the same idea. There's the base guanine, which is also a purine, so there are 2 rings there. And if it was the monophosphate version, we'd have guanosine monophosphate, or GMP. Now, just for the record, you're probably thinking this. If you had an adenine-based nucleotide that had 3 phosphate groups on it, it would be adenosine triphosphate, right? Or ATP, which you're all probably familiar with. And this would be GTP, which you may be familiar with. There are certain signaling proteins known as G proteins that make use of GTP and its affiliated species. So I guess we should mention one other thing just to be really correct. It's not really the topic of this section, but RNA and DNA have certain similarities, and RNA nucleotides and DNA nucleotides have certain similarities and certain differences. The main difference, if this were RNA, would be that, remember, this is deoxy, right? So this is important, I think, even though this is not a lecture about RNA. But RNA nucleotides, really the only difference between them, I shouldn't say the only, but the main difference between them, is that at the 2' carbon, they have an OH group instead of an H. And so that sugar, if it has that OH there on the 2' is called ribose. When you deoxy it, look what you do. You deoxy it, get rid of the oxygen. That's all deoxy really means. The reason I bring that up is because there's RNA version of, for example, ATP, and there's the, quote, DNA version of ATP. So if you really want to be careful, we're dealing with DNA nucleotides here, right? You can call it DATP, or deoxyadenosine triphosphate. The reason I bring it up is because the regular ATP, that is the energy-carrying molecule of the cell, which again, you see a situation where a molecule that plays a role in genetics is also playing a role in energetics. That's not deoxy. That's sort of the RNA version of the nucleotide. Okay? Sorry if this is getting too far into this, but I think it's a good level for you guys to know. So you could do the same with DGTP versus regular GTP. It's important if you want to be complete. Now that we get the idea, we can look at the pyrimidines and probably not go into quite as much detail. So the base thymine, here it is, has one ring only. That's why it's a pyrimidine. And since there's only one phosphate group here, this would be thymidine monophosphate, or TMP. The trinucleotide form of it would be TTP, or of course DTTP if we want it to be very specific. And over here we have cytosine, and the nucleotide that contains it and one phosphate group would be cytidine monophosphate, CMP. So, without going into craziness, you can have DCTP, CTP. I guess, if you had the CMP, you could even write DCMP, but that's getting a little bit much at this point. So, I guess I should have said this right off the bat, but I think you're getting the fact that, so here we have the sugar. There are 1, 2, 3, 4, 5 carbons in that sugar. It's a pentose, this deoxyribose. They call them prime, by the way. A lot of people have asked me this, is because when biochemists number the carbons in a whole nucleotide, they start with the base. So you're less likely to hear about the carbon numbers in the base, but they're numbered 1, 2, 3, etc. The ones in the sugar are numbered 1 prime, 2 prime, 3 prime. So if you hear prime, you know that you're talking about a carbon in the sugar as opposed to a carbon in the base. So there. That's the sugar part, that's the base part up there, and then here's the phosphate group. So each nucleotide, as we said, is a little bit complex, having 3 components, but it functions pretty much as a unit when it's time to get into a chain. Now here's a summary of a bunch of things we've said. I mentioned to you that nucleotides tend to exist as the triphosphate versions when they're not incorporated into a chain, and thus this is just a summary of the very specific denotations of DNA nucleotides. So we have DATP, or I wrote adenosine triphosphate, but technically it would be deoxyadenosine triphosphate, and you can just go down the list: D-guanosine triphosphate, D-thymidine triphosphate, deoxycytidine triphosphate. So this is the way often you'll encounter the nucleotides, and if they're not in a DNA chain. Now, all this stuff was pretty much known before Watson and Crick, and you may think that they discovered everything. I'm not taking away from their achievement at all, but they had a lot to work with going into it. So they said, look, we have this polymer of 4 nucleotides. Furthermore, they gathered Watson and Crick, and again, this is not a knock on them at all. They were more theoretical scientists than experimental scientists. So they actually asked around chemists, people doing all sorts of things. What did they know about nucleotides and how could they put this puzzle together? It's really what they did, is brilliantly put a lot of pieces of a puzzle together. For example, there's some controversy about this part. That's not my job to tell you about, but Rosalind Franklin was a scientist who was doing X-ray diffraction on DNA. This is something that you'd have to be an expert to actually interpret the data, but she had done a lot of X-ray diffraction data implying that there was something helical about the way DNA was arranged. And without going into a long story about it, her boss, Maurice Wilkins, gave Watson and Crick the data, and some people think that Rosalind Franklin sort of got ripped off, not getting the credit for it. Sadly, she also died shortly thereafter. But anyway, so they had X-ray diffraction data. This guy Chargaff had come up with the idea that was very important. He wasn't sure why, but in a bulk analysis of DNA, he said that the amount of A always equaled the amount of T, and the amount of C always equaled the amount of G, very specifically. There was a theory before this that DNA was a repeating polymer of just A, G, C, T. I forget the order, but in other words, just no variation, just over and over and over again, the 4 nucleotides, one after the other. Chargaff's results really disproved this, but he wasn't able to say why A equals T and C equals G. Watson and Crick took all this, put it together, and figured out a model of how DNA actually exists in nature in the cell. And it does explain how it meets those 3 conditions that we mentioned for, preconditions of the genetic molecule. So now famously, they envisioned what they called a double helix, which we'll take a look at in a moment. Some people now call it a twisted ladder conformation, etc. But the bottom line here is, in their model, 2 chains of nucleotides, and I really guess I should say polynucleotides, meaning they were very long strands. They were intertwined around each other, thus the double helix, and the nucleotides were joined together by so-called phosphodiester bonds. And for the reason that we just said, it gives each chain an intrinsic directionality, meaning 5' to 3'. When they put it all together, what they saw, or what they proposed, I guess, was that the two strands are held together by hydrogen bonds, specific hydrogen bonds, between the bases. And because you'd have to really be a biochemist to see this, or maybe you can just go back and look and see, but there are only appropriate spots on G and C to be able to make very stable hydrogen bonds, and A and T. So at least at this level, there are some weird things that can go on in molecular genetics, but pretty much G and C make one type of so-called base pair, and A and T make the other type. As far as we're concerned, there's no such thing as, for example, AC base pair. By the way, one of the things they thought of first is that there has to be one pyrimidine, sorry, that would be the smaller one, and one purine in order to span the distance across the helix. So that was clear. You'll notice that that's a purine, that's a pyrimidine, purine, pyrimidine. So they knew that that should be true, but furthermore, the specificity of GC and AT is very important. So in summary, they said that the double helix consists of two anti-parallel strands. Anti-parallel simply means that if one strand is running in the 5' to 3' direction this way, the other strand is running in the 5' to 3' direction the other way. So it's almost like they're upside down in relation to each other. And we'll see the picture in a minute, but if that wasn't the case, the bases would not be facing each other, which they have to be in order to engage in this pair bonding, this base pairing via hydrogen bonds. So not only is that the case, but it's all twisted into a helical, very definite helical shape, and that conformation is known now to provide the lowest free energy. In other words, it's the most stable way that this whole thing can go together. It's also consistent with the X-ray diffraction data that they had that showed this general helical nature to things. I don't know if you'd have to memorize this, but if you think of the helix sort of as a cylinder, the diameter is 2 nanometers, which is rather small, and there are about 10 base pairs for each complete turn of the helix. So let's take a look at one. This is sort of an isolated, out of context thing, but think of this as here's one nucleotide, right? In this case, it's a base that's a T here, a thymidine. Notice that it's attached to the 1' carbon of the sugar. Now, over here we see that this is the idea of the anti-parallelism. Granted, it's only one base, but we have a 5' free over here and a 3' there. It's sort of upside down on the other side. It has to be, because this base that's attached to the 1' carbon of the sugar has to be facing inward. And it's an A, and we know that because this you have to memorize, unless, just memorize it. A always has to go with T, G always has to go with C. So in the case of AT, there are two stable hydrogen bonds that can be formed. In the case of a GC bond, there are 3 stable hydrogen bonds that can be formed. Sometimes this has importance in how stable the helix is, the melting temperature, but you don't need to worry about that right now. So hopefully you understand the way that these two nucleotides are interacting. Let's put it in the context of a very large, long DNA molecule. And guys, they can be literally millions of bases in length. All right, so I like this picture a lot because it sort of shows the double helix, but then it opens it up at the end. So here's 2 nanometers in diameter here. One complete turn, if you look in there, there should be 10 base pairs, is 3.4 nanometers. And what they're saying here is that each individual base pair has a width, I guess you would say, of 0.34 nanometers. I suspect you wouldn't have to know those exact figures, but you never know. It's up to your professor. What we really want to do, though, is look down here and see how it opens up at the end. So what they've done is sort of undone the helix at the bottom part here, what we've done. And we're seeing that, number one, so if this is a 3' end, right? We're running 3' to 5' in this direction. That means this one is doing the opposite. It's running 3' to 5', and there you see the 5' end of that strand. So these strands can be extremely long. What's drawn sort of as a ribbon here is often known as, I should write it, well, it already is written here, the sugar phosphate backbone. The sugar phosphate backbone is very important, but it's structurally unchanging. It goes that way throughout the length of the entire molecule. What's different, and we started out by saying this, is that which base is attached. So if it's a C, we know that we're going to see a G over on that side. And while this is highly diagrammatic, you see that, I hope you can see, if there were two purines or two pyrimidines there, the combination of them would be either too wide or too narrow to span the length of this helix, which we know to be 2 nanometers. So whether it's CG or whether it's GC, they always have to go together. And you see here an AT bond and TA bond, basically the same thing. So one way or another, we have to get to the bottom of here is that how does this meet the 3 conditions? So we had said, first of all, 2 of them are sort of similar. There has to be a way for variation to occur, right? Well, it turns out, erase these in here, that the order of the bases, G, C, T, A, G, C, T, whatever it is, I mean, there are almost infinite possibilities in terms of what type of information this can encode. So the different bases going along in when I say non-predictable, I mean it's not just like it's always GCTA, GCTA, GCTA. It can be any sequence. There are some constraints we'll talk about later, but that's how the information is encoded. You may remember we said that a DVD is encoded simply with strings of zeros and ones, right? Lots of them. Well, we got 4 subunits here, and it's enough to encode all of the biological information in the known universe. That's pretty massive. Also, we had talked about mutation and the capability of change and variation, its role in variation. So we're not going to see why right now, but every now and then mistakes get made and bases can change their chemistry a little bit. So suffice it to say that every now and then, and that sounds a bit unscientific, but it's true, during replication usually one base can change to another one, and that's the basis of variation. Also, we're going to have a whole lecture about it next time, but probably the most powerful reason that Watson and Crick thought they were correct in this model is, we won't go into the details, but the last thing was that DNA had to be able to replicate itself, right? And while they didn't go into the details, they immediately saw conceptually a way that DNA could copy itself. And this is so important. So you'll notice that if this model is correct, which it is, if you know the sequence of one of the strands because of the base pairing rules, you automatically know the sequence of the other strand. Isn't that true? And vice versa. So what they envisioned was, they said that if you did unzip the double helix, as it were, as it is here, each side could act as a template to automatically make a copy of its partner strand because of the base pairing rules. So that's extremely important, and DNA replication will be the topic of our next session. I'll see you then.